Distinguished guests, ladies and gentlemen, members of the membership of the Faculty of Pharmaceutical Medicine, it gives me great pleasure to introduce Professor Sir Michael Rawlins, who is going to give a lecture on medicines regulation and the role of the MHRA. Now, I should add that Professor Sir Michael Rawlins is an extraordinarily distinguished clinical researcher, certainly within the UK, certainly globally. And it was my pleasure earlier this afternoon to make him an honorary fellow of the faculty. In fact, he's been a fellow of the faculty since 1989, and as I explained this afternoon, it was long overdue for us to recognize his extraordinary contribution and his achievements over quite a few years. I won't say specifically how many. He was trained as a physician in London at St. Thomas's originally and at the Hammersmith Hospital. At a fairly tender age, he became professor of medicine at the University of Newcastle. He is the current chair of the MHRA, but he will certainly be known to many of you as the driving force behind NICE in this country, where he served with distinction for, I think is it 14 years? Yes, it was 14 years. He's been chair of the Commission on Human Medicines. He's done an extraordinary amount of work in conjunction with his interests, which have also included Huntington's disease. His career has simply been extraordinary, and it would really I would take up the time of this lecture simply giving you a long list of his publications, recommendations, achievements that span his career. So Mike, over to you in terms of medicines regulation and the role of the MHRA.
Thank you, Keith, for that too kind introduction. It is a very great honour to be invited to deliver the faculty's annual lecture. I hope I meet your expectations, even if I've messed up the very first slide. Those of you who realise I've called the MHRA by the wrong name. It's the Medicines and Healthcare Products Regulatory Agency. I only noticed it 2 seconds ago. I was sitting there looking at it. There are many facets to the role of the MHRA in medicines regulation, and I will focus on just a few. As I'm sure most of you know, the MHRA is comprised of 3 centres. The largest, with about 800 staff, is the Regulatory Centre. The National Institute for Biological Standards and Control, based at South Mimms near Potters Bar, has about 350 staff. The Clinical Research Data Link, CPRD, has about 60 staff. It emerged from the General Practice Research Database a few years ago. I will talk in some detail of the activities and future plans of both the Regulatory Centre and CPRD, especially as they relate to medicinal products, but less about NIBSC. I should emphasise, though, that NIBSC plays a very important role in maintaining the nation's health, and my neglect in this talk is really for reasons of time. The Regulatory Centre has a wide range of responsibilities. These include the regulation of medicines, of which there are around 15,000 on the market at the moment, as well as the regulation of devices, with more than 500,000 currently being marketed. The Centre also has responsibilities for regulating blood and blood banks, as well as herbal products, homeopathic remedies, and the annual production of the British Pharmacopoeia, now celebrating 150 years of publication. The Centre also has responsibilities for inspection, standards and enforcement, as well as a policy function, particularly in negotiations relating to relevant EU legislation. As far as the regulation of medicines is concerned, the Regulatory Centre has responsibilities for licensing, for vigilance and risk assessment, and as I have already explained, for inspection standards and enforcement. A very important licensing function is the grant of clinical trials authorization, and substantial changes are afoot. The current arrangements for clinical trials in the EU were laid down in the Clinical Trials Directive of 2001. By general agreement, it was a disaster. Indeed, the Commission itself admitted it to be the most heavily criticised directive of all time. It lacked clarity about what constitutes a clinical trial, and so was variably implemented and interpreted across the EU. It was disproportionate and attempted to apply the same standards to a simple volunteer study of a widely used product such as paracetamol as to a new monoclonal antibody for the treatment of cancer. This one-size-fits-all approach was a disaster and was probably at least partially responsible for a decline in clinical trial activity across the EU. In the UK, we applied the Clinical Trials Directive to the letter. Just let me give you one example. Some years ago, a small group of palliative care physicians wanted to find out whether high-dose morphine and high-dose diamorphine in late-stage cancer was associated with cognitive impairment. The question was a perfectly reasonable one. Are patients on high-dose opiates competent, for example, to make a will? The oncologist therefore planned to compare cognitive function in patients with late-stage cancer on high-dose opiates and those on conventional doses. When they applied for a clinical trial authorization, they were told, 1, they needed a GMP certificate for a preparation they were already using from regulated sources as routine therapy. 2, under GCP rules, the opiates would have to be stored at +4 degrees centigrade in a fridge with a temperature monitor, despite the fact that opiates are normally kept at room temperature in the ward drug cupboard. As controlled drugs, this meant that the fridge would have to be locked and screwed to the wall. Finally, they were told they would have to obtain indemnity cover, which was going to cost thousands of pounds. At that stage, not surprisingly, they gave up. Furthermore, and to make matters worse, some of the GCP inspectors were overzealous in their scrutiny of trial sites. One said to an NHS director at an NHS trust, "Gotcha," when he found some minor fault. Revised arrangements for the authorization of clinical trials were agreed by the EU in 2014. It is a regulation. The room for inconsistent implementation should be minimal. It takes a proportionate, risk-based approach. There is to be a single EU portal with a single application package and harmonized procedures enabling multi-state trials to be authorized without bureaucratic delay. The bad news, though, is that it cannot be implemented before 2017 because the electronic portal needs to be built and rigorously tested before it goes live and is implemented. The good news is that the MHRA is already implementing the spirit of the new regulation. Moreover, there has been a sea change in the attitudes of the GCP inspectors, who, by all accounts, are now helpful and instructive on their visits to study sites. The Regulatory Centre is also taking a keen interest in emerging novel trial designs, adaptive pathways, so-called basket trials, umbrella trials, Mendelian randomization, step wedge trials, and its close relative ring trials. First, then, adaptive pathways. The idea in adaptive pathways is that there are 2 steps in the demonstration of efficacy and safety. The first is the experimental step, during which the effect size is examined in conventional phase 2 type studies. If the product appears to show significant efficacy and safety, the sponsor seeks conditional marketing authorization. If granted, the sponsor is expected to follow treated patients to assess whether the promise is fulfilled during the observational period. Note that during this observational trial period, there is no placebo. If the initial promise is fulfilled, then the sponsor would be granted full marketing authorization. This arrangement will obviously only work for conditions where the natural history of the disease is reasonably predictable. It'll also require prescribers to use the product solely within its license or indications and to cooperate fully in the collection of data. The advantages of the arrangement are obvious. Patients will have access to new products at an earlier stage in their life cycle, and sponsors can start obtaining some return on their investment. Furthermore, by reducing a product's development time, this arrangement should also mean that the acquisition costs are substantially reduced. The EMA is currently undertaking pilot studies to assess whether adaptive pathways are efficient, safe, and effective. Basket trials, it's an unfortunate term really, because we talk about, in Europe, we talk about, well, in Britain, basket cases, but they don't have basket cases in the United States, where much of this has come from. Basket trials are a new and evolving form of clinical trial design. It is predicated on the hypothesis that the presence of a molecular target will predict the response to a targeted therapy independent of tumor site or histology. The success of the basket trial depends in large part on the strength of the data linking the target and targeted therapy. For this trial design to work, two key conditions must be met. The tumor must be dependent on the target pathway, and the targeted therapy must reliably inhibit the target. There are statistical considerations in this novel clinical trial design that will continue to play a central role. It does, though, offer the possibility that we are entering an era where diseases are characterized by their molecular profile rather than their site and histopathology. The approach is being particularly used in oncology. For instance, recent reports from the Sloan Kettering Institute in New York suggest that the BRAF mutation, common in malignant melanoma, can be observed in a small proportion of patients with acute leukemia. And those expressing the BRAF mutation, a striking clinical response has been observed when given targeted treatment. Acute leukemia is a relatively uncommon malignancy, and the proportion expressing the BRAF mutation appears to be less than 5%. The difficulty in undertaking a conventional randomized controlled trial for such a population of patients is obvious. Nor might basket trials be confined to oncology. It has been suggested, for example, that the technique could be used in the development of new antimicrobial agents. Unlike basket studies, where investigators test the effect of a single drug on a single mutation in a variety of cancer types, umbrella trials are designed to test the impact of different drugs on different mutations in a single type of cancer. So we have one disease, say breast cancer, with several molecular subtypes and several therapies. This design allows validation of a strategy based on a mixture of biomarkers and drugs. Again, there will be scientific and technical issues in the analysis and interpretation of the results of umbrella trials, but regulatory authorities will need to be flexible in their assessment of the results. Mendelian randomization has been used in epidemiological research for more than 20 years, but it is starting to be used in interventional studies. The approach works like this. A group of patients with a particular phenotype, say type 2 diabetes, are treated with a specific product. At the end of the study, the response is compared in patients with a specific genotype compared to the response in patients lacking the specific treatment. Clearly, this approach will only work for conditions where similar phenotypes occur with different genotypes. The analysis of the data is, though, very similar to that of a conventional randomized controlled trial. It should be relatively straightforward. Step wedge trials can be used to investigate efficacy and safety in individual clusters. In this instance, for example, I'm presuming there are 4 clusters of patients. At the start of the study, step 1 cluster out of the 4 is treated. The remaining clusters act as controls. At an appropriate time interval, a second cluster, step 2, is added. At step 3, a third cluster is included, and at step 4, the final cluster is treated. In this way, all the participants receive the active treatment, and effects in treated clusters are compared to ones awaiting treatment. So-called ring trials are very similar to step wedge trials. The design was used in assessing the efficacy of a novel Ebola vaccine in Guinea earlier this year. The particular vaccine studied was a recombinant stomatitis virus-based vaccine expressing a surface glycoprotein of Zaire Ebola virus. After laboratory confirmed cases of a confirmation of a new case, clusters of all contacts, as well as the contacts of contacts, were identified. All members of each cluster were randomly offered either immediate vaccination or delayed by 21 days vaccination. As you can see, there was no case of Ebola virus disease amongst the more than 4,000 individuals undergoing immediate vaccination. But 16 confirmed cases of the just over 3,500 individuals in the delayed vaccination group. For this, the vaccine efficacy was estimated to be 100%, with confidence intervals ranging from 74 to 100%. Clearly, no vaccine can ever produce 100% efficacy, but this study does demonstrate that this particular vaccine appears to have a high degree of protection against Ebola EB virus. Another issue currently taxing the regulatory centre is the wide availability of counterfeit medicines on the internet. These alleged products include treatments for a wide variety of conditions, with fake Viagra being a particularly prominent product. Not only are these treatments ineffective, but they can sometimes have fatal effects. You may recall reading about a young woman who purchased a slimming aid online and died. She'd been sold unknowingly dinitrophenol, which uncouples oxidative phosphorylation in mitochondria. Many other hazardous substances are available online, including products prepared from human plasma, which, when the MHRA's enforcement team went into the premises of the manufacturer, were labelled not for human use. Other products, including antidepressants, steroids, and antibiotics, can be also purchased online from unscrupulous vendors. One of the MHRA's enforcement officers, a former police officer, told me the other day that the people selling counterfeit medicines are the same people who are selling controlled drugs, such as cocaine and cannabis, and who are also the same people involved in human trafficking. In other words, it's organised crime. Counterfeit and falsified medicines are defined as medicinal products with a false representation of the product's identity, its name and composition, its source, its country of origin, and its history, especially that relating to the distribution channels being used. As you may be aware, the MHRA has special powers to take down the websites promoting falsified medicines, as well as powers of entry into premises where they are held. This year alone, the MHRA has taken down 1,600 websites selling falsified medicines. Problems, though, remain. First, our powers are confined to websites based on servers in the UK, and we're unable to remove those that are offshore. Second, no sooner do we take them down, and they pop up again a day or two later on a new and different site. Third, counterfeit medicines can also be purchased from what is called the dark web or dark net. More of that in a moment. To tackle the international sales of counterfeit medicines, a program called Operation Pangaea is run for a week every year. For those of you who didn't read Mods and Greats at Oxford, Pangaea is the hypothetical landmass that allegedly existed when all the world's continents were joined together about 200 to 300 million years ago. The MHRA is very actively involved in Operation Pangaea, which is coordinated by Interpol with 115 participating countries. The activities targeted in Operation Pangaea include internet service providers, payment systems, and delivery services. This year, Operation Pangaea took place during the second week of August. In just 7 days, over 20 million fake and illicit medicines were seized, with an estimated street value of over £50 million. There were over 150 arrests, more than 400 investigations were launched, and nearly 2,500 websites taken offline. As far as it goes, this year's Operation Pangaea was a success. Nevertheless, illicit medicines, as I've already indicated, are available on the dark net. I lived such a sheltered life that the dark net or dark web was something of which I was completely unaware until I became chairman of the MHRA. The dark net is what is called an overlay network that can only be accessed using non-standard encrypted communication systems via an anonymized series of connections. I'm reliably told that almost anything and everything can be purchased on the dark net, including guns, hard drugs, and even hitmen. Apparently, the sites for hitmen have customer satisfaction ratings like TripAdvisor. The mind boggles. 0 out of 5 missed, 5 out of 5 headshot. I don't know. I have no idea. I did actually Google the darknet a few days ago in preparation for this lecture, and I was looking at it, and I suddenly thought, I think I better stop this. The boys and girls from Cheltenham will be watching me and parking their lorry outside. The darknet also has sites for the purchase of pharmaceutical products. A recent review by Bloomberg Business claimed that it was possible to obtain almost any pharmaceutical from the darknet. And major pharmaceutical companies have become extremely worried by this trend. Indeed, they're employing specialists to try and take these sites down. Here's a site on the darknet selling flunitrazepam, Rohypnol, or Roofies, as they're sometimes called. As well as being an effective hypnotic, Roofies are a date rape drug, although the UK most commonly used rape drug is actually gamma-hydroxybutyrate, or GHB. And before you conclude, despite my protestations, that I am indeed a surfer of the darknet, this advertisement was taken from the Bloomberg article I mentioned a moment or two ago. But taking action against the supply side of counterfeit medicines on the internet is not enough. Action needs to be taken to reduce the demand side of the sales of illicit medicines on the internet. The Falsified Medicines Directive introduces a common logo across the EU. Its presence on a website that is legally selling medicines confirms that the online retailer is authorized to sell medicines to the public. And here is the logo they can display. The MHRA is taking a pragmatic approach to the implementation of the new EU common logo regime. The application portal went live in mid-June this year, and online retailers have been provided with the logo since the beginning of July. The agency is supporting online retailers as they work to meet the new legislative requirements, and we wouldn't expect them to cease trading during the assessment period, provided they had already submitted an application to us. Starting in the new year, we plan an extensive publicity campaign so that consumers are aware of the dangers of buying drugs on the internet from sales not displaying the logo. This is the one and probably only occasion in my lifetime that I want to appear on the front page of the Daily Mail. The second of the MHRA's centres is the Clinical Practice Research Data Link, or CPRD as it's commonly known. As I've already indicated, it is based on the previous General Practice Research Database, GPRD, started in the late 1980s, but with a substantial number of additional functionalities. CPRD is primarily a database of primary care records that include data on mortality, morbidity, and on prescriptions. There are a total of 19 million people's data on CPRD, with around 5 million patients that are currently live. CPRD is now linked to other data sets such as the HES data, the Hospital Episode Statistics data, and can be linked to others. And CPRD has become such a rich source of research data and information that nearly 2,000 peer-reviewed papers have been published over the 25 years based on it. CPRD has 3 uses. First, it has been and continues to be extensively used for pharmacoepidemiological research by the MHRA itself, by academic investigators, and by pharmaceutical companies. I'll show you an example of one such study in a moment. And CPRD is now being used to examine the safety of devices. Secondly, CPRD is also starting to be used for intervention research. 2 feasibility investigations have shown this is possible, and the agency has recently contracted to undertake a formal clinical trial using CPRD. But thirdly, CPRD can be used for broader types of epidemiological research, and again, I will give you an example shortly. Here, though, is an example of CPRD resolving an extremely important public health problem. You will recall some years ago, this man, Andrew Wakefield, claimed to have shown a causal association between MMR vaccination and pervasive developmental disorders, including autism. His claims were widely reported in the lay press, and although his evidence was flimsy in the extreme, MMR vaccination rates tumbled as frightened parents failed to be reassured about its safety. Using CPRD, Professor Liam Smeeth at the London School of Hygiene and Tropical Medicine identified almost 1,300 children with pervasive developmental disorders, looked at their immunisation history, and compared this with nearly 4,500 controls. Here I've summarised his findings. Whether you look at all children with pervasive developmental disorders, those with autism only, or those with autism spectrum disorder, there is not the slightest suggestion of an increased risk as shown by these adjusted odds ratios and their associated 95% confidence intervals. This forest plot shows Smeeth's finding in relation to other, but smaller case control studies examining the same issue. None of the studies individually indicated an increased risk, and the overall pooled odds ratio confirms this. There is no indication that MMR immunisation is associated with pervasive developmental disorders. CPRD is a critical tool for studying drug safety. But as I've already mentioned, it is capable of answering other questions related to population to health. For reasons too complicated to explain now, I have an interest in Huntington's disease. George Huntington, like his father and grandfather before him, was a family doctor in East Hampton on Long Island. His description, published in 1872, of what was then known as the Magrams or hereditary career, has become a classic. His eponymous disease is a progressive neurodegenerative condition, usually, though not invariably, presenting in midlife. It is characterized by worsening career and increasing cognitive impairment. And patients die between 10 and 20 years after the onset of symptoms. Huntington's disease is inherited as an autosomal dominant trait, which means that each child of a parent with Huntington's disease knows that they have a 50% chance of developing it themselves. It is a relatively rare disease, and there's been considerable uncertainty about its overall prevalence in the UK and whether there are geographical differences across the country. So, using CPRD and with colleagues from the London School of Hygiene and Tropical Medicine, I've been examining the prevalence of Huntington's between 1990, the start of reliable data from CPRD, and 2010. Our study showed that over this 20-year time frame, the UK prevalence of Huntington's has increased from just over 5 per 100,000 of the population to more than 12 per 100,000. We also showed that within the UK, there are remarkable geographic differences in the prevalence of Huntington's. This shows the average prevalence between 1990 and 2010 in each of the 12 regions of the UK. In the majority, the prevalence is between 8 and 9 per 100,000, but there are some marked differences between London, with a prevalence of around 4 and 5, and Scotland and the northeast of England, with prevalence rates 3 times higher. Although, again, we can surmise possible reasons, we do not really know why the regional differences exist. But why has the prevalence increased? One explanation is that the incidence has risen, perhaps because of improved diagnostic techniques or perhaps increasing mutation rates. But it might be artifactual. There has been for many years a shame associated with having Huntington's disease in the family. And GPs might have been reluctant to enter the diagnosis in patients' records. Indeed, even today, many Huntington's families ask their doctors to avoid including a diagnosis of HD in death certificates. Thirdly, it could be due to an increase in survival, either associated with the general increase in longevity that has occurred over the past 20 years and/or improved symptomatic treatments. The rise in prevalence actually certainly can't be attributed to an increase in incidence. This shows incidence rates in 7-year bands between 1990 and 2010, again based on data from CPRD with no evidence of any significant change. Not only can we exclude a rise in incidence causing the rise in prevalence, but it appears that the shame hypothesis is unlikely too. If it was contributing to the increased prevalence, we should have also seen a rise in incidence. So we are left with the possibility that survival is increased, something we are currently exploring using CPRD. And finally, the National Institute for Biological Standards and Control, NIBSC. It's based in South Mimms with a staff, as I said, of around 350 scientists and has a number of critical functions. First, it provides over 95% of the world's biological standards. It is also the official medicines control laboratory for biological medicines, including vaccines. It has the ability to rapidly respond to adverse incidents and was intimately involved in the Ebola outbreak in a variety of different ways. It has recently established an advanced therapy division to provide appropriate standards for cell therapy. And it has also undertaken a wide variety of research projects and provides opportunities for research students. It is a massive asset to both the MHRA and the UK as a whole. So in conclusion, the synergies between the MHRA's 3 centres are critical to the agency's mission. Although each has its own areas of responsibility and expertise, as well as ambitious plans for the future, the whole is far greater than the sum of the parts. The MHRA is a great organisation and one of which I am proud to be part. Thank you very much indeed. Thank you.